Artificial Intelligence Dr. www.abcpharma.net/English. User question: what are the specific molecular mechanisms by which different macronutrients, carbohydrates, fats, proteins, influence satiety signaling pathways, and how can this knowledge be leveraged to develop more effective dietary interventions for weight loss? GPT-4o answer: different macronutrients, carbohydrates, fats, and proteins influence satiety signaling pathways through various molecular mechanisms. Understanding these mechanisms can help develop more effective dietary interventions for weight loss. Here's how each macronutrient affects satiety and the associated signaling pathways. 1. Carbohydrates. Glucose and insulin response. 1. Mechanism. Carbohydrate consumption leads to an increase in blood glucose levels, which stimulates insulin secretion from the pancreas. 2. Satiety signaling. Insulin acts on the central nervous system, CNS, to promote satiety by interacting with insulin receptors in the hypothalamus. 2. It also enhances the uptake of glucose into cells, reducing blood glucose levels and signaling fullness. 3. Glycemic index. High glycemic index, GI, carbohydrates cause rapid spikes and drops in blood glucose and insulin, leading to short-term satiety. 3. Low GI carbohydrates provide a more sustained release of glucose, leading to longer-lasting satiety. Gut hormones. 1. GLP-1, glucagon-like peptide 1. Carbohydrates stimulate the release of GLP-1 from the intestine, which enhances insulin secretion, slows gastric emptying, and promotes feelings of fullness. 2. PYY, peptide YY. Carbohydrates also increase the release of PYY, which acts on the hypothalamus to reduce appetite and prolong satiety. 2. Fats. Energy density and gastric emptying. 1. Mechanism. Fats are energy-dense and slow down gastric emptying, leading to prolonged digestion and sustained release of nutrients. 2. Satiety signaling. The delayed gastric emptying increases the time food stays in the stomach, contributing to prolonged satiety. Hormonal responses. 1. CCK, cholecystokinin. Fats stimulate the release of CCK from the small intestine. 1. CCK acts on the vagus nerve and the hypothalamus to promote satiety and reduce food intake. 2. Leptin. Fats contribute to adipose tissue expansion, which increases the secretion of leptin. 2. Leptin acts on the hypothalamus to regulate energy balance and suppress appetite. Fatty acid oxidation. 1. Mechanism. The oxidation of fatty acids provides a steady energy supply, which can signal the brain to reduce hunger. 2. Satiety signaling. Fatty acids can directly act on the hypothalamus to influence satiety pathways and reduce food intake. 3. Proteins. Amino acid sensing and hormonal response. 1. Mechanism. Protein consumption leads to the release of amino acids into the bloodstream, which can be sensed by the brain. 2. Satiety signaling. Amino acids stimulate the release of hormones like GLP-1 and PYY, enhancing satiety and reducing appetite. Hormonal modulation. 1. GLP-1 and PYY. Similar to carbohydrates, proteins increase the secretion of GLP-1 and PYY, which slow gastric emptying, enhance insulin secretion, and promote fullness. 2. Ghrelin. Protein intake suppresses the release of ghrelin, a hormone that stimulates hunger, thereby reducing appetite. Neurotransmitter synthesis. 1. Mechanism. Amino acids from proteins are precursors for neurotransmitters such as serotonin and dopamine, which play roles in mood and satiety. 2. Satiety signaling. Increased synthesis of these neurotransmitters can enhance feelings of well-being and reduce the desire to eat. Leveraging this knowledge for dietary interventions. Balanced macronutrient distribution. 1. Strategy. Develop diets that include a balanced distribution of carbohydrates, fats, and proteins to leverage their combined effects on satiety. 2. Example. Meals with low GI carbohydrates, healthy fats, and high-quality proteins can provide sustained energy release and prolonged satiety. Protein-rich diets. 1. Strategy. Emphasize higher protein intake to enhance satiety through hormonal responses and amino acid sensing. 2. Example. Including lean meats, fish, legumes, and dairy in meals to reduce hunger and promote weight loss. Healthy fat inclusion. 1. Strategy. Incorporate healthy fats that slow gastric emptying and stimulate satiety hormones. 2. Example. Using sources of unsaturated fats like avocados, nuts, seeds, and olive oil. Low GI carbohydrates. 1. Strategy. Focus on low GI carbohydrates to avoid rapid spikes in blood glucose and insulin. 2. Example. Whole grains, vegetables, and legumes that provide sustained energy and satiety. Timing and meal composition. 1. Strategy. Optimize meal timing and composition to maximize the satiety effects of each macronutrient. 2. Example. Combining proteins and healthy fats with each meal and snack to maintain satiety throughout the day. Conclusion. Understanding the specific molecular mechanisms by which carbohydrates, fats, and proteins influence satiety signaling pathways allows for the development of more effective dietary interventions for weight loss. By leveraging these mechanisms, balanced and personalized dietary plans can be created to enhance satiety, reduce overall calorie intake, and promote sustainable weight loss.